Good morning. It is my e-presentation on study of prophylactic action of linseed oil against acetaminophen induced oxidative stress and liver damage in albino rat. Myself, Dr. Brij Mohan Singh, Associate Professor, Department of Zoology, Sri Kalyan Government Girls PG College, Sikar, Rajasthan. Abstract. In this investigation, the protection conferred by the linseed oil against acetaminophen induced oxidative stress liver damage has been evaluated in adult albino rat. Hepatotoxicity has been evaluated within the indication of elevated level of enzymatic activity of aspartate aminotransferase, alanine aminotransferase, alkaline phosphatase and bilirubin level of in acetaminophen administered rat. Co-treatment with linseed oil significantly restored the elevated level of hepatic serum markers AST and ALP, alanine aminotransferase, alkaline phosphatase and aspartate aminotransferase. This may be the result from the cellular leakage due to the peroxidase damage of the membrane. Significant reduction in antioxidant activities of superoxide dismutase, glutathione catalase and glutathione phosphatase were obtained as compared to the control values. At the normal condition, the body defense mechanism against oxidative stress marked by the endogenous antioxidant enzyme such as SOD, GSH, catalase and glutathione phosphatase prevent cell damages induced by the free radicals. The protective effect of linseed oil are dose and tissue dependent. Due to differences in the inherent susceptibility of each tissue in rat, remarkable elevation in the bilirubin level and reduced level of cholesterol in comparison to the control group of rat were found in the study. Thus, the augmented antioxidant enzyme level in the liver tissue of rat treated with linseed oil justifies the antioxidant property of the oil. Introduction. The plant derived product have been attracted a lot of attention of many researchers today who find their medicinal potential for the treatment of various diseases. Nowadays, most of the people believe solely on the plant products as medicine to cure the diseases. Linseed oil is an antioxidant substance which is derived from the dried ripened seeds of flax plant. It contains 52 to 63 alpha linolenic acid and beneficial effect in reducing inflammation leading to atherosclerosis. Linseed oil may reduce cardiovascular risk through platelet function and inflammation. It has positive effect on femur bone mineral content and bone mineral density and lumbar vertebrae. Jean Rao found that the high dose of linseed oil can also delay in memory cancer. Acetaminophen, in other words, N-acetyl aminophenol paracetamol is an important antipyretic, analgesic and effective therapeutic medicine which high dose induces liver toxicity, which is usually characterized by the chest pain, vomiting, diarrhea and sometimes shock in mammals. Oxidative stress is attributed to the hepatotoxicity in mammals. Liver injury by the overdose of acetaminophen is caused by the increased production of reactive oxygen species, antioxidant depletion and cytokine regulation. Material and methods. Linseed oil has been given orally to the rat. Different concentration of linseed oil were given to the rat for the selection of optimum dose in terms of milligram per kilogram body weight. After supplementation of 15 days, sublethal concentration of pro-oxidant acetaminophen has been injected to the animal. The survivability of the body weight were checked up to 15 days. The dose of acetaminophen which produced 50% mortality within 30 days was estimated. The group which showed the maximum survivability and healthier condition were selected for the study. Swiss albino mice have been divided into 3 groups. One is control group, were not received any other treatment. Second is the administered group of dose of acetaminophen and third is the co-exposure of acetaminophen plus linseed oil. Rat were sacrificed at various interval ranging from 1 to 15 days. Immediately after the exsanguinations and liver were removed, rinsed, cleaned, weighed. A small portion of liver were kept frozen in order to analyze antioxidant enzyme activity and estimation of bilirubin cholesterol level. Mice liver were studied time to time for qualitative and quantitative parameters, color, weight of liver, histopathological and biochemical analysis of standard methods in pathological laboratories. All serum samples were processed for the determination of enzymatic activity of aspartate aminotransferase, alanine aminotransferase and alkaline phosphatase using a spectrophotometric method. Antioxidant activity of superoxide dismutase and glutathione and catalase and glutathione phosphatase were determined in the serum sample with ELISA kit. To estimate the bilirubin and cholesterol level, a random access chemistry analyzer RA1000 was used. At least 3 random visual fields from each animal were scored in a blended manner by 2 expert pathologists. Results and discussion. Exposure of rat to the acetaminophen caused a significant elevation in the hepatic serum markers, alanine aminotransferase, aspartate aminotransferase, alkaline phosphatase respectively in comparison with the control group. This may result from the cellular leakage of these enzymes into the cytoplasmic circulation and peroxidative damage to the membrane produced evidence in hepatotoxicity. Aminoacetaminophen toxicity causes the corresponding destruction of hepatocytes and thus resulting in an elevation of serum enzymes. Co-treatment with the linseed oil significantly restored and elevated level of hepatic serum markers. These levels affect the basic antioxidant status of the cell. Thus, compounds that ameliorative oxidative stress can cause an improvement on oxidative damage to the liver as shown in figure 3a, b, c. These biochemical restoration may be due to the inhibitory effect of the linseed oil on cytochrome P450 and promotion of acetaminophen glucuronidation. In the present study, 250 mg per kilogram acetaminophen caused acute liver injury was characterized by the increased serum activity. A remarkable reduction in antioxidant activities of superoxide dismutase, GSH, catalase and GPX by these percentages were obtained as compared to the control group. The protective effect of the linseed oil are dose and tissue dependent, probably because the differences in the inherent susceptibility of each tissue in rat. Remarkable elevation in bilirubin level and cholesterol level down were found during the study period. While the bilirubin level was lowered and cholesterol level were higher in comparison when compared with the acetaminophen treated group is attributed to the hepatocellular damage as shown in figure 3b. Linseed supplementation in group 3rd has returned to the level of bilirubin and total cholesterol at almost the same level as the control group. The bilirubin is a conventional indicator of liver disease. The mounting evidence of the previous studies Das and Viswas has supports what we found in the present study, that is acetaminophen induced toxicity evoked elevated bilirubin level, depleted GSH level after administration of linseed oil, and deleterious effect of acetaminophen were significantly ameliorated in the comparison to the acetaminophen group. Overall, these ameliorating potential of linseed oil confirm the hepatoprotective effect of the oil. This is the table which showing the result of alanine aminotransferase activity, aspartate aminotransferase activities, and alkaline phosphatase, superoxide dismutase, GSH, catalase, and GPX and bilirubin cholesterol activities. In the acetaminophen administered group, this is the percentage of administered group, and this percentage is the combined or ameliorating effect of the acetaminophen and linseed oil. This is the graph showing different activities of various graphs, and this graph is showing the various activities. These are the figures showing the hepatocytes liver conclusion. The study highlights the hepatoprotective effect of the linseed oil improving the defense mechanism of the liver oxidative stress. The hepatoprotective effect of this